Okay, so after having just talked about cellular respiration, we're going to move away from that component and transfer into a different metabolic process known as photosynthesis. Kind of just a disclaimer, I apologize in advance if there are any disruptions due to my cat Figaro. He is having a difficult time dealing with daylight savings and the fact that he has to wait about an hour more for his food. So I decided to feed him an hour early so he would shut up, and hopefully the beast has been distracted and will not make any interruptions, but cannot say anything for sure. So without further ado, move into photosynthesis. I like to say in general that life on Earth is solar powered, right? Because photosynthesis nourishes most of the living world, whether that is direct relationship or more of an indirect sustenance per se. Now, as far as the type of organisms we have, there are autotrophs. Autotrophs are your self-feeders and your biological producers. And then the other organisms are heterotrophs, which are your consumers. Autotrophs can sustain themselves without eating anything that's derived from other living material or other living organisms because they're able to produce their own energy. They're able to produce organic molecules from carbon dioxide. More specifically, when we talk about plants, we refer to them as photoautotrophs because they use light as the source of energy to synthesize organic compounds. And as such, they are the ultimate source of organic compounds that are required by heterotrophs for energy. Almost all your heterotrophs are dependent on whether it's partial dependence or complete dependence on photoautotrophs for food and oxygen. Oxygen will be a byproduct of photosynthetic processes. So let's actually talk about photosynthesis itself. What is photosynthesis? Photosynthesis in general is this multi-step process that will require sunlight, carbon dioxide, and water. Those three components will serve as substrate. Now, what about the products of photosynthesis? Well, this process is going to release oxygen and a carbohydrate molecule. More specifically, that carbohydrate molecule that's produced is called glyceraldehyde 3-phosphate, or G3P. This is a simple carbohydrate that will then subsequently be converted or can be subsequently converted into other carbohydrates such as glucose, sucrose, etc. Now, the process of photosynthesis, it's believed that it evolved as a way to store the energy in solar radiation, as these high energy electrons in the carbon-carbon bonds of carbohydrate molecules. Right, so that energy from solar radiation is harnessed and it's stored as potential energy in, again, those carbon-carbon bonds of your carbohydrates. And that potential energy can then be obtained by other organisms when those bonds are broken. So you have the switch from potential to kinetic energy. Now, the point where students start running into trouble or issues with photosynthesis is kind of right about now, because they start confusing the processes, reactants, and products of photosynthesis with those of cellular respiration. And that's understandable, especially when you are just learning about these two components for the first time, but it's also understandable because there are several commonalities. For example, both will involve redox reactions. Both involve the production of ATP. And essentially, photosynthesis and cellular respiration are opposites of one another. I'll show you a comparison chart in a moment, but they literally are opposite processes. It's much more complicated than that in reality, but just for the sake of simplicity, so you don't kind of go mad, we'll just say that they're essentially opposite reactions of one another. So plants, they'll use both photosynthesis and cellular respiration. Animal cells will only use cell respiration because we do not have chloroplasts. Now, when we're going through this lecture, kind of, again, going back to the techniques that I used back in my younger years when I was going through schooling way back when, what worked best for me as far as preventing confusion was thinking of both of these processes kind of as being in completely separate boxes on completely opposite sides of the room, right? So you have a little file for cell respiration and a separate file for photosynthesis, right? They're both metabolic processes, but they are very different. Now, I don't want you to forget what I've taught you or what you have learned about cellular respiration. But for the moment, I kind of want you to put that information away in its own little folder, in its own little file cabinet. And don't think about it. Think right now only of photosynthesis. So this is going back to that point that I made, that you can think of photosynthesis and cell respiration as being opposite reactions. In photosynthesis, you have the production and storage of energy. In cell respiration, you have the release of energy. So in photosynthesis, you're storing that energy. By the means of cellular respiration, you're releasing that energy that had been stored. So think of them as cycles, right? So you're storing or you're getting your energy through photosynthesis. That energy is then, that was originally stored, is now going to be released by cellular respiration. In photosynthesis, all right, the process uses carbon dioxide and it produces oxygen. That oxygen that's produced is going to be used by cellular respiration to produce carbon dioxide. And guess what this carbon dioxide is going to be used for? That's right, it's going to be used in photosynthesis, again, to produce more oxygen that will be used in cellular respiration to produce more carbon dioxide. Right? This is just your air cycle or carbon dioxide cycle. Water. So photosynthesis will use water. It also produces water as a byproduct to a very small degree. Most of your water will be produced in cellular respiration. So again, cyclic, right? This produces or this uses water that's produced by cellular respiration. So glucose, right? So in photosynthesis, photosynthesis is going to produce glucose from energy sources. That glucose that's produced is going to be burned or utilized in cellular respiration. Photosynthesis will take place in your chloroplasts, and it's going to require light, mostly for the first phase of photosynthesis. The second phase, or the Calvin cycle, can occur either in light or dark. We call it a dark reaction. We'll talk about that in a moment. But here you can really see how, again, or where I'm coming from when I say that they are essentially opposite reactions, but you can also see this cyclic relationship between the two. So again, both different processes, but they are similar in many ways, which is why there's so much confusion that arises. But thinking of photosynthesis as its own entity, again, as more of kind of creating a lot of the starting materials for cellular respiration, and then thinking of cellular respiration as producing the starting material for, or the material that can be recycled and used for photosynthesis. Again, you think of them in their own separate categories, but remember that relationship between them, it can help you better separate the two processes and keep them from becoming confused with one another. All right, so photosynthesis itself is an anabolic chemical reaction, or an anabolic process. As an anabolic process, it's an endergonic reaction, and it also contains redox reactions. Now, as with any other type of reaction that involves energy, so any sort of energetic reaction, photosynthesis will involve energy transformations. In particular, of importance is what type of transformation is occurring. So in photosynthesis, you have the transformation of light energy into chemical energy. Now, the amount of energy that's transformed is dependent on the amount of light that is captured or photosynthesized. In other words, it's dependent on photosynthetic rate. Now, plants will adjust their photosynthetic rate depending on their external environment. So based on the environment that they're living in, they will modify their photosynthetic rate in accordance. And by doing that, they can alter the biochemical composition of their leaves so that they can adjust, again, their photosynthetic rate based on the resources that are available. Do they need to increase photosynthetic rates because they are in more of a shaded area, or is there enough sun exposure that they can actually decrease the photosynthetic rate a little bit because they are receiving so many photons of light? Fun fact, the plant species that has the highest rates of photosynthesis is bamboo. So, fun little fact that you can share over casual drinks at the bar. Alrighty, so the kind of net equation or net reaction for photosynthesis is the one that's pictured here. Again, the actual product here, it says the product of photosynthesis is a sugar. Yes, it is, but more specific, that actual direct product is a 3-carbon sugar, again, called your glyceraldehyde 3-phosphate, or G3P. And that can be used, again, to synthesize other carbohydrates like glucose. So as I said before, photosynthesis is an anabolic process or anabolic reaction. As such, it's also an endergonic reaction. And if you remember on our talk about energy and metabolism, endergonic reactions are those that require energy, right? Energy is entering the system. And the reason for this is because in photosynthesis, you're going from smaller molecules to larger molecules of a carbohydrate, right? So that is a synthesis reaction or an anabolic process. in order to do that, again, you need to incorporate energy. Energy is needed to be put into the system. Now, photosynthesis is also a redox reaction, or it's comprised of many different redox reactions, where you have one molecule that's being oxidized and the other is being reduced. Right? So one is losing electrons, that's oxidized, while the other is gaining electrons or being reduced. If you forget some of these concepts as far as the redox reactions, go back to that cellular respiration PowerPoint and take a peek. Now, in your photosynthetic reactions, right, thinking again of them as these redox reactions, water is split and electrons are transferred with those protons from a molecule of water to carbon dioxide, reducing it to a sugar. So carbon dioxide, right, here's your molecule of carbon dioxide that you obtain from the air, is reduced into a sugar. The water molecules that are part of your reactants for photosynthesis, they will be oxidized to form molecules of oxygen, right? So these go through your oxidation, your carbon dioxide goes through reduction, right? So again, carbon dioxide is reduced to produce or form sugar. Water is going to be oxidized into oxygen. Now, kind of an interesting tidbit of history here. One of the first clues as to the mechanisms of photosynthesis came from the discovery that oxygen given off by plants comes from water, not carbon dioxide. Right? So when you initially look at this reaction, a lot of people are going to be like, okay, the oxygen is coming from the carbon dioxide. And that's not true. The oxygen, again, that's produced in photosynthesis is coming from your water. And this fact was confirmed back in the 1960s through the use of isotopes. Researchers specifically used oxygen 18, which is a heavy isotope, and they used it as a tracer to follow the fate of these oxygen atoms during photosynthesis. So what they did was they labeled either molecules of carbon dioxide or molecules of water with isotope, and then kind of equally disperse them between reactions. So some reactions had one, others had the other. Some had the radio-labeled carbon dioxide, some had the radio-labeled water. And the researchers found that the oxygen 18 label appeared in the oxygen from photosynthesis only when water was that source of the tracer. So those reactions that had a water molecule that was loaded with that heavy isotope tracer produced oxygen or yielded oxygen through photosynthesis that contained that tracer or also contained that label. So that research allowed for the understanding that, again, these oxygen molecules that are produced in photosynthesis, again, are coming originally from those water molecules that are used as reactants or incorporated at the beginning of the reaction. So that the hydrogen from water, when you actually have the split of the water molecule, the hydrogen is going and is being incorporated into the sugar, while that oxygen is released into the atmosphere. Now, kind of thinking in a different direction, right? So here you see, in order to go from here to here, so from reactants to your products, you need light, right? So because the electrons increase in potential energy as they move from water to sugar, you need energy. So the process requires energy. Again, that energy is going to come from light. This here is more of the full equation for photosynthesis. Again, you see water being used as one of the reactants, but it also appears on both sides of the equation. So there's a little bit of water that is produced, but this is the net equation, right? So you'll incorporate 6 molecules of carbon dioxide plus 6 molecules of water and light. Again, light is going to be your energy source to allow for the production or yield of carbohydrates or a sugar molecule and 6 molecules of oxygen. Okay, so before we go into the actual kind of nitty-gritty what's happening per se in the steps of photosynthesis, I want to take a few minutes to go over the anatomy of leaves, since they are pretty important to this process. They're pretty important because leaves happen to be the major site of photosynthesis for most of your plants. There are about 500,000 chloroplasts per square millimeter of leaf surface. The green coloration of a leaf is derived from chlorophyll, which is the green pigment of chloroplasts. Now, more specifically, the process of photosynthesis will occur in this middle layer that's called the parenchyma, or it's also known as the mesophyll or mesophyll. Your mesophyll is composed of 2 separate types of parenchyma. So you have your palisades parenchyma and the spongy parenchyma. So this is together, again, they form your general parenchyma or the mesophyll. Your mesophyll is where chloroplasts and chloroplast containing cells are largely found. Now, oxygen will exit and carbon dioxide will enter leaves through these microscopic pores that are called stomata. You can think of them as these little gateways. The stomata, you can see them right here. So this is not the stoma. This picture is incorrect. This entity itself is kind of a little Cheerio looking thing. That is the stomata, this word here, right? So stomata serve as the sites of gas exchange in leaves. They're typically located on the underside of the leaf in an effort to minimize water loss through transpiration. Now, each stomata is flanked by guard cells. And these guard cells will regulate the opening and closing of the stomata by swelling or shrinking in response to osmotic changes. So based on the osmotic stance of the plant, whether it is turgid or is flaccid, again, depending on its osmotic stance. So whether it's in a hypotonic or a hypertonic environment, it will, or those guard cells will either swell or shrink to regulate the opening and closing of the stomata. Now, going more into, or kind of more in depth in your plants. So again, here's your stomata. That's where oxygen will exit and carbon dioxide will enter the leaf. It's also where you can have the loss of water from transcription. Inside you have your mesophyll. Again, that's composed of your palisade parenchyma, right, which is kind of this linear stack, per se, of parenchyma cells, and then your spongy parenchyma. Now, taking a close-up look at one of these mesophyll cells, so looking at one of these guys, and you see these little green blobs, those are called chloroplasts, right? So taking a deeper look at your chloroplasts, there can be anywhere from 30 to 40 chloroplasts in one mesophyll cell. The chloroplasts you can think of as these chemical factories that are powered by the sun. Each chloroplast has two membranes, right? So they have a double membrane that surrounds this central aqueous space, per se. That aqueous space is called the stroma. Within the stroma, there's this elaborate network of interconnected membranous sacs that are called thylakoids. These are, again, these stacked disc-shaped structures, right? So here's a little thylakoid. Here's one here, here, and here. Now the thylakoid membrane encloses an internal space that's called the thylakoid space or the thylakoid lumen. And then most of these thylakoids will be stacked on top of one another to form columns that kind of look like these stacks of IHOP pancakes. Right? Now these stacks are called the granium. Right? So they're just stacks of thylakoids. So where is the chlorophyll located? Well, the chlorophyll is located in the thylakoids. The thylakoids in your chloroplasts transform light energy into chemical energy of ATP and NADPH. All right, so let's actually, now that we know about the anatomy, go into the actual stages or nitty-gritties of the processes of photosynthesis. Photosynthesis is composed of two sequential stages. The first being light reactions, or your light-dependent reactions, and the second being the Calvin cycle, which are light-independent reactions, or your dark reactions. Now, the light reactions take place in the thylakoid, and they serve as the conversion of light energy, or your solar energy, into chemical energy. So energy from sun or from some sunlight is absorbed by the chlorophyll and used to make ATP and NADPH. Right? So again, it converts energy into stored chemical energy. So your light reactions are the photo part of photosynthesis. The Calvin cycle, or again, your light-independent reactions, this will take place in the stroma. So again, that aqueous space of your chloroplast. And it uses chemical energy that is harvested or created during the light reactions to incorporate carbon dioxide from the atmosphere into sugar molecules. So again, it uses those products of the light-dependent reactions to make sugar from carbon dioxide. Again, that sugar molecule will be your glyceraldehyde 3-phosphate, or G3P. So the Calvin cycle is the synthesis part of photosynthesis. So we'll start with the light reactions. Now, when we talk about the light reactions or your light dependent reactions, they require, well, light, hence their name. Light is a form of electromagnetic energy or radiation. And like other forms of electromagnetic energy, it travels in the form of waves. Now, when we talk about waves, the distance between peaks of waves, right? So the distance from here to here is called a wavelength, right? So the distance between the peaks of waves are wavelengths. Now, the entire range of radiation that exists is known as the electromagnetic spectrum. Now, the segment of the spectrum that's important to us is visible light. Oh, thank you, computer, for that notification. Visible light encompasses the range of the spectrum from 380 to 750 nanometers in wavelength. Now, even though light itself travels as a wave, it has many properties of what is called a photon. A photon is a discrete particle of light. You can kind of think of it as like a little marble of light energy. Now, the amount of energy that's packed into that marble, right? The amount of energy that's packed into a photon is inversely related to wavelength. So the shorter the wavelength of light, the more energy that is packed into a photon. Now, your sun will radiate essentially or almost a full electromagnetic spectrum, right? So again, all of these will be emitted from the sun. However, the atmosphere itself, you can think of as acting as a screen door. It's going to filter out most of the wavelengths. Visible light is the only one that passes through in significant quantities. And again, it's of importance to us because visible light is what drives photosynthesis. Now, compounds that absorb light, these are called pigments. So when light meets some sort of matter, right, there's one of three things that can happen, or multiple things that can happen at once. Light may either be reflected, absorbed, or transmitted. Now, with pigments, that light is absorbed. The wavelengths that are absorbed by a pigment will disappear. Right? They are not portrayed as the color of the object. The color that's actually observed is the aspect of visible light that is being reflected. Right? So let's take our little leaf friend, for example. A leaf appears green because of chlorophyll, which is the dominant pigment. And chlorophyll absorbs red and violet or blue light. So because it's absorbing light from that area of the spectra, it transmits or reflects green light. So the light that's reflected is the color that we observe. Therefore, that's why we see leaves as being green. Now, there are three major types of photosynthetic pigments that are contained in chloroplasts. You have chlorophyll A, chlorophyll B, and carotenoids. Now, they will all differ in absorption spectra, meaning that they absorb different areas of that visible light spectra. Chlorophyll A participates directly in the light reactions. It's really awesome at absorbing red and violet wavelengths, but it stinks at absorbing green, right? Instead, it reflects green. And this is the vital light capturing pigment, again, that will participate directly in your light reactions. Our second little friend is chlorophyll B. So chlorophyll B absorbs from either end of the spectrum, right? So absorbs blue and orange light, or light in that region of the of those wavelengths. It will reflect yellow-green light. Now it has a slight structural difference from chlorophyll A. So the porphyrin head, or porphyrin head of your chlorophyll A pigment will have a CH3 group here, whereas in chlorophyll B you'll have carbon with a hydroxyl group. So that slight structural difference allows them to have differences in their absorption. So it allows them to absorb wavelengths of light at different regions of the spectra. Figaro, stop. Now your carotenoids, these are kind of like an accessory pigment. You find these oftentimes in fruit. And in fruit, they're used as advertisements to attract pollinators or seed dispersers. When we're talking about kind of their function in leaves, they're really neat because your carotenoids provide this element of photoprotection. Right? So the carotenoids are hydrocarbons, and as such, they will absorb and dissipate excessive light energy that would otherwise damage the chlorophyll or interact with oxygen to form these harmful reactive oxidative molecules. So again, it allows for photoprotection, or allows or protects from damage because of too much light exposure. Over here you have this absorbance spectra that shows the wavelengths of light best absorbed by these different types of chloroplast pigments. Now, many photosynthetic organisms will use a mixture of pigments, and by doing so, they're able to absorb energy from a wider range of wavelengths. So more efficient with photosynthesis because they can absorb more energy from a wider array or a higher range of different wavelengths. So going back to this slide here, here's the structure of one of these pigment molecules. Each pigment has that porphyrin ring, which is the light absorbing head of the molecule. And then they have a hydrocarbon tail. That tail will interact with the hydrophobic parts of proteins that are inside thylakoid membranes. Now your pigments, they will be arranged in what's called a photosystem, which is essentially this collection of pigment molecules that are embedded in a protein. All right, so again, the first process of photosynthesis is going to be your light-dependent reactions. So they're using visible light, or photons of light are entering the system to allow for this reaction to take place. In the process, a water molecule is split, so that will provide a source of electrons and protons, while also giving off oxygen as a byproduct. Light that's absorbed by the chlorophyll will drive that transfer of electrons and protons from that water molecule to a different molecule called nicotinamide adenine dinucleotide phosphate, which is this little guy here, your NADP plus. So those electrons from the split of water are transferred to this molecule here. And in that process, or once that molecule gains those electrons, it's transformed or converted into NADPH. Figaro, get out of the basket. Stop playing with the fake chicken. No, I said get out of the basket. Oh, dear Lord. Do you want to be locked in the room? Yeah, I didn't think so. Okay. So electrons transferred. You have a redox reaction to create NADPH. Now ATP is also going to be created. ATP will be generated through chemiosmosis through a process called photophosphorylation. If you remember from cell respiration, we talked about the generation of ATP that it can occur through 3 different processes. You can either have substrate level oxidation, you can have oxidative phosphorylation, or photophosphorylation. Photophosphorylation is only going to occur in photosynthesis, more specifically, kind of right smack in the middle of your light-dependent reactions. In photophosphorylation, you have the transfer of electrons because of light, or kind of fueled by light energy, to allow for the synthesis of ATP from ADP and an inorganic phosphate. White! Oh my goodness gracious! Such a needy little kitty cat. So light energy in your light-dependent reactions is converted into chemical energy in the form of NADPH and ATP. All right, so going into the nitty-gritties of what's happening in your light reactions. When a chlorophyll pigment or any other pigment absorbs light, or a photon of light, an electron is boosted to an excited state. So it's elevated to an electron shell where it has more potential energy. When your electron is in a normal shell, right, so before it does that bounce, it's said to be in ground state, right? So your pigment at that point would be in ground state. The absorption of a photon boosts electrons to higher energy state, so you have a higher energy electron shell that it moves to, and in that process, or at that point, your pigment is said to also move into an excited state, right? So when it gains, again, these electrons from light, your pigments move from ground state to that excited higher energy state. Now, each pigment or each compound absorbs only certain photons, right? They will only absorb photons that correspond to a specific wavelength, which is why each pigment has a unique absorption spectrum, right? That's why each pigment has a different peak absorption, or a different wavelength at which they best absorb. They're specific to a specific wavelength. Now, when you have that transition in your electrons, right, it goes from ground state to this excited state following the absorption of a photon of light. At this excited state, it is very unstable. So the electron won't stay there for long. That excited electron will jump back down or drop back down, usually after the absorption of another photon of light, back to ground state, or a ground state orbital. That usually occurs in about 1/1000th of a second. Now that excess energy in this process, so when it moves from excited back to ground state, is released as heat. Some pigments can also release additional photons of light that produce fluorescence. You can observe this by exciting chlorophyll. Computer, shut up. Exciting chlorophyll with ultraviolet light, and it will glow. Now, the conversion of light into chemical energy occurs or takes place in a multi-protein complex that is called a photosystem. Now, there are two photosystems embedded in the thylakoid membrane that cooperate together in light-dependent reactions. And they're called, very simply, photosystem II and photosystem I. Now, they are named in order of their discovery, not in order of their functioning in photosynthesis. Photosystem II is actually the first photosystem to operate in your light-dependent reactions. They both have the same basic structure. They'll differ on the basis of what they oxidize, so the source of that low energy supply, and what they reduce, or the place to which they deliver their energized electrons. But again, they both have the same basic structure. A photosystem is composed of a protein complex that's called a reaction center complex, which is kind of this component within this core. You can kind of think of this as almost looking like a little red pepper. All right, here are those little bulges on the outside of the pepper. That middle area where you see the stem, think of that as being this internal core or a reaction center complex. The reaction center complex includes two special chlorophyll A molecules, right? So it has a little pair of chlorophyll A molecules, as well as a number of these light harvesting complexes, right? So the light harvesting complexes will surround this reaction center complex that contains your chlorophyll A pair. Each light harvesting complex, right, so your little bulges of the red pepper, consist of pigment molecules. These pigment molecules are bound to certain proteins. Now, the number and variety of protein molecules enable a photosystem to harvest light over a larger surface area, as well as a larger portion or range of the electromagnetic spectrum, or the visible light spectrum. Now, together, these complexes your light harvesting complexes will act as antenna for the reaction center complex. Right? These light harvesting complexes are going to receive light, right? And that light, again, is going to fuel a series of reactions that will be harnessed by that reaction center complex. More specifically, your light harvesting complex will pass energy from sunlight onto the reaction center. Now we're going to talk about that actual process kind of individually for each photosystem, right? So starting with the first photosystem functioning in photosynthesis, or in, sorry, your light-dependent reaction of photosynthesis. That first photosystem is going to be photosystem 2. Right, so this guy here. Photosystem 2 has a reaction center chlorophyll A molecule or chlorophyll A pair known as P680. And what that means is that it has an absorption peak, right? This chlorophyll pigment has an absorption peak at 680 nanometers, right? So we say the reaction center of photosystem 2 is P680. Right? So photosystem 2, you have the entrance or absorption of a photon of light, which strikes a pigment molecule that is located in the light harvesting complex, or one of the complexes. When that photon is absorbed, it will boost one of the electrons in this pigment that absorbs it to a higher energy level, right? And again, that little electron is going to go from ground state to excited state. And then because it's in that unstable excited state, it will quickly drop back down to ground state. Again, that occurs in about a billionth of a second. When it falls back to ground state, an electron in a neighboring pigment molecule is simultaneously raised to an excited state. Right? So this one goes to excited state, then falls from excited back to ground. As it's falling from excited state back to ground state, it's exciting a different electron in a neighboring pigment. Now this process will continue, right? So it kind of bounces from one pigment to another until it reaches the P680 pair of chlorophyll A molecules in the reaction center complex. Once it reaches this pair, one of the electrons in the P680 is going to be excited, right? So looking at this pigment here, right? So this pigment is going to have an electron that goes from ground to excited state, and then it falls from excited back to ground state. As it's falling back to ground state, it's exciting electrons in P680, right? So one of these electrons in P680 is excited to a higher energy state. Now that excited electron, that electron that's in that higher or that higher energy state or the excited state, right? It's going to be this high energy electron. That's going to be delivered by P680 to what's called a primary electron acceptor. So the primary acceptor, or your primary electron acceptor, is going to take or pick up that high energy electron from P680. And what do you know? You have a redox reaction occur here, right? So your P680 drops off that high energy electron, and in that process, P680 is oxidized to P680 plus, right? It's P680 with a +1 charge. And then your primary acceptor is reduced, right, because it has gained those electrons. Now, P680 plus is the strongest oxidizing agent known, right? So in this state, where it's missing an electron, it becomes very, very reactive. And essentially has now this electron hole that needs to be filled. So what's going to fill that hole? What's going to replace that missing electron? Well, that missing electron is going to be replaced by extracting low energy electrons from water, right? You're going to have this enzyme that comes along and it breaks water or a molecule of water into 2 electrons, atomic oxygen, which is your half O2, and 2 protons. And that enzyme will take those electrons from that broken down water molecule and supply them to P680 plus to fill that hole. So your P680 plus oxidizes water, and by doing so, it is reduced back to P680. Right, so you go from P680 plus to P680 by gaining the electrons or taking the electrons from water. The other components, kind of wondering what happens to the rest of the components of this water molecule. Well, the protons, they'll be released into the thylakoid space. Right? So they're going to be released in here outside of your membrane, right? So here's the thylakoid membrane. Outside over here is the stroma. Inside here you have your thylakoid lumen or thylakoid space. So that's where your protons are going to move to. The oxygen atom, right, this atomic oxygen, will combine with another atomic oxygen or oxygen atom to form O2. So that's what's happening again at photosystem 2, right? So this is how, or that was how we get back to P680. What happens now to the electron that was gained at this primary electron acceptor? Well, your photoexcited electrons, as we can call them, they pass from the primary electron acceptor of photosystem 2 all the way to photosystem 1 via an electron transport chain. Oh no, it's another electron transport chain. It's okay, this one is really easy. It's not like the crazy one in cellular respiration. The electron transport chain that lies between these 2 photosystems is made up of 3 components. You have an electron carrier. I think I have a better picture. Here we go. So you have an electron carrier called plastoquinone or PQ, a cytochrome complex, and a protein called plastocyanin or PC. Now the cytochrome complex is kind of the main star or queen of this transport chain. This complex will transfer electrons from your carrier molecule, PQ, to the protein, PC. And in that process, it also enables the transfer of protons across the thylakoid membrane, as well as, again, that transfer of electrons from photosystem 2 to photosystem 1. Now, as your electrons are moving through this electron transport chain, you can think of them as falling down an energy gradient or an energy waterfall per se. Essentially, what you have occurring here is this exergonic fall of electrons, each one falling to a lower energy level. And as these electrons, again, they were originally high energy, each passage they're going to fall to a lower energy level, so that when they are picked up by photosystem 1, you're now picking up these low energy electrons. But as they're dropping to these lower energy levels, that's providing energy that is harnessed. And what do you think that might be harnessed for? Well, that energy is harnessed and used for the synthesis of ATP. Right? So what's happening is, as electrons pass through this cytochrome complex, your protons or your hydrogen ions are pumped into the thylakoid space or your thylakoid lumen. And that contributes into the creation of a proton gradient that will be used to fuel chemiosmosis or the chemiosmotic synthesis of ATP. So that's going to, or your generation of ATP, again, is going to occur kind of midpoint of your light-dependent reaction. It's going to occur again right during that electron transport chain, and you're going to more specifically have the light reaction generating ATP through a process called photophosphorylation, and using light or light energy to drive the synthesis of ATP through chemiosmosis. So what's happening is as you move down the electron transport chain, right, and even at this part here where you have the split of the water molecule, you have the pumping of protons into the thylakoid space. So protons are building up inside the thylakoid lumen or thylakoid space to create this concentration gradient or this electrochemical gradient, right? Different regions of charges across a selectively permeable membrane. Now, what will happen is you'll have the passive movement or your diffusion of your hydrogen ions or your protons from high concentration, right, which is going to be in the thylakoid space or thylakoid lumen, to low concentration, right? And that's low concentration is going to be in the stroma. Now that diffusion is going to occur through this specialized structure that's in the thylakoid membrane called ATP synthase, right? Again, just like cellular respiration. When they rush through this specialized protein channel, they will be used to create energy, right? So the energy released by this proton stream through ATP synthase allows the protein to attach a third phosphate group to ADP and thereby forms or synthesizes a molecule of ATP. Right, and again, this is through photophosphorylation because you're using light energy to drive this reaction. All right, so again, now we're coming out of your electron transport chain. So in the electron transport chain, we had the generation of ATP as well as the transfer or transport of these electrons between photosystem 2 to photosystem 1. So let's talk about what actually happens in photosystem 1, right? So at photosystem 1, another photon of light is absorbed, and it's absorbed by your little pigment molecules in these light harvesting complexes of this photosystem. And just like in photosystem 2, that light energy is transferred via little pigments that are in the light harvesting complexes. And they'll be transferred between these pigments until reaching the reaction center complex, where at that time, those electrons will excite a pair of P700 chlorophyll A molecules. Again, those molecules will be located in the reaction center complex. So this chlorophyll A molecule is different from your photosystem II one. Right? Again, these are P700 chlorophyll A molecules, meaning that they have an absorption peak at 700 nanometers. And these guys had an absorption peak at 680. So the electrons reach your P700. That causes your P700 to move to a higher energy state or an excited state. Right? So they're carrying a higher energy electron now. And that photoexcited electron is transferred to your photosystem 1's primary electron acceptor. Right? So again, you basically same thing that's happening or that had happened in photosystem 2. This primary acceptor is going to take those high energy electrons, and in the process that creates an electron hole in P700. Right? So P700 has now been oxidized to P700 plus. And in this oxidized form, P700 plus can act as an electron acceptor. So it will kind of fall back to the end of this reaction center complex, and in that P700 plus form, it can accept an electron that reaches the bottom of the electron transport chain from photosystem 2. Right? So again, it's a recycling process. Right? So it will be oxidized. So P700 will be oxidized to P700 plus when it drops off its high energy electron at the primary electron acceptor. And then it will be reduced back to P700 when it picks up or is moved into that excited state when it gains the electrons that are basically dropped off from photosystem 2. Now, once those high energy electrons are delivered to the primary acceptor, they will be kind of passed in a series of redox reactions from the primary electron acceptor of photosystem 1 down a second electron transport chain that moves through this protein here, FD, which is known as ferredoxin. Now, important note, this electron transport chain that's happening after photosystem 1, it does not create a proton gradient, and it does not produce ATP. The importance of this chain is the synthesis or formation of NADPH. Right? That's the only thing it's going to do. So in this chain, again, you have your primary electron acceptor, it's going to pass off electrons or transfer electrons to this protein structure, this protein being ferredoxin. Now, an enzyme called NADP plus reductase catalyzes the transfer of the electrons from your ferredoxin to your NADP plus. In order for kind of the reaction to occur here, you need 2 electrons. Right, so 2 electrons are required for the reduction of NADP plus by NADP reductase into NADPH. This process will also remove 1 proton from the stroma. Now, this molecule that's produced, your NADPH, again, what you have going on here, this is the reduced form. So NADP plus gains electrons and gains protons to form to create the reduced form NADPH, right? So NADP plus is reduced by this enzyme to form, voila, NADPH. Now, where's this guy going to go? Well, this molecule will be used for the Calvin cycle, right? So that's where we're going to go next. But before we do, you can think of the kind of linear flow of electrons during the light reaction between your photosystems, kind of using this mechanical analogy. You have a photon of light comes in, and that triggers the movement of electrons from a low energy state to a high energy state in photosystem 2. And then that high energy electron will be kind of transported down this little slide here. And in the process, it catalyzes the turning of this mill, and that mill is going to generate ATP. And then the electrons will move, as they move down this slope, they go down in energy level until they hit photosystem 1, where you have another photon of light enter in that allows that low energy electron to bounce back up to an excited state, where that excited or high energy electron is used to create NADPH. All right, so now we can move into the second process. The second process is the Calvin cycle. The Calvin cycle occurs in the stroma, and we also refer to it as dark reactions, right? Because it doesn't specifically require light. We also refer to as light-independent reactions. So in the Calvin cycle, or your light-independent reactions, the chemical energy that was harvested during the light-dependent reactions will actually drive the assembly of sugar molecules. And these sugar molecules are assembled from carbon dioxide. So this is, again, that synthesis component of photosynthesis. This is where the production of sugar actually occurs. And as I said, it doesn't directly require light. Right? So light itself is not directly used as a reactant, but it does require the products of those light-dependent reactions in order to function. So it will use the chemical energy of ATP and NADPH to reduce carbon dioxide into sugar. Again, the specific sugar that will be immediately produced is going to be glyceraldehyde-free phosphate, or G3P. And the Calvin cycle is similar to the citric acid cycle, in that the starting material will be regenerated after several steps of the cycle. It differs from citric acid cycle in that your Calvin cycle is an anabolic process, right? Krebs is catabolic. You're breaking down these carbohydrates into different materials to obtain the resources to allow for the reduction of different high energy carriers so that you can form NADH or FADH2. Here in the Calvin cycle, it's anabolic because you're building carbohydrates from smaller molecules and in the process consuming energy. You're not creating energy. So carbon will enter the cycle as carbon dioxide. It leaves as a sugar. Again, that sugar product, more specifically, is a 3-carbon sugar called glyceraldehyde 3-phosphate. All right, so let's talk about what actually goes on, right? The nitty-gritty steps. So the Calvin cycle, or your light independent reaction, is going to use that NADPH and ATP that was created from the light dependent reaction to fix atmospheric carbon dioxide onto different carbohydrates. Now, the carbon dioxide that's used will enter leaves through the stomata, and then it will diffuse over a short distance through some intercellular spaces until it reaches the mesophyll cells. Once it's at the mesophyll cells or in the mesophyll cells, the carbon dioxide diffuses into the stroma of the chloroplast. And again, that stroma is the site of these dark reactions, or Calvin cycle. Now, each turn of the Calvin cycle fixes one carbon. In order to have the synthesis of one glyceraldehyde 3-phosphate molecule, or G3P molecule, the cycle must take place 3 times, or you need to have the fixation of 3 molecules of carbon dioxide. Now you can think of this molecule of G3P that's produced as being half a molecule of glucose, right? So if you wanted to create a full molecule of glucose from the Calvin cycle, that cycle would need to take place 6 times. So you need 6 cycles to occur and the fixation of 6 carbon dioxide molecules to create 1 molecule of glucose. Here we're just creating G3P, which can then be combined with others or altered to form other carbohydrates. So in order to have 1 molecule of G3P formed, we need 3 cycles in order to fix 3 molecules of carbon dioxide. Now, the Calvin cycle is going to do this through 3 phases, right? In other words, it's the Calvin cycle is comprised of 3 different phases. These phases being carbon fixation, reduction, and regeneration. Now, carbon fixation is the first stage. In this phase, carbon is fixed onto organic molecules. So carbon dioxide enters. It's added to a 5-carbon sugar that's called ribulose biphosphate, or RUBP. Now, this reaction is catalyzed by this cool little enzyme here called ribisco. Another name for it is RUBP carboxylase. So this enzyme is going to create a 6-carbon intermediate by adding that carbon dioxide to the 5-carbon sugar. The intermediate that's created is very unstable, and it splits in half to form 2 molecules of 3-phosphoglycerate. In other words, 2 molecules of 3-PGA. for each carbon dioxide. Right? So for one carbon dioxide molecule, it creates 2 of these 3 PGA guys. But because this will have 3 cycles, or you have the fixation of 3 carbon dioxide molecules, in total, you'll have the synthesis of 6 molecules of 3 PGA. Right? So a total of 6 molecules of 3 PGA are produced in carbon fixation. From here, we move into the second phase or stage, which is reduction. Essentially, what's happening here is this molecule is being reduced twice. Right? So stage 2, reduction, well, you have reduction happen twice in this stage. Good way to remember it. So during reduction, each of these 3 PGA molecules receives a phosphate group from ATP to form a molecule that's called 1,3-biphosphoglycerate. Again, that's a redox reaction, your first one, which involves the gain of electrons by 3 PGA to form your 1,3-biphosphoglycerate, and the loss of electrons from ATP. So ATP here is oxidized to ADP. Following that, you have your second redox reaction. So a pair of electrons from NADPH reduces each 1,3-biphosphoglycerate into G3P. Right? So your NADPH is oxidized back to NAD+. Where do you think this NADP+ will go? Right, it's going to be cycled back to your light dependent. But here, again, it's reduced. So it's reduced to, sorry, not reduced, it's oxidized to your NADP+. While it's oxidized, or because it's oxidized, it's giving away its electrons or protons, and that's picked up by the 1,3-biphosphoglycerate to form G3P. So 1,3-biphosphoglycerate is reduced to G3P. Now here, for each carbon dioxide molecule, you'll have a total of 2 G3P made. So 2 G3P per cycle. But we're talking about 3 molecules of carbon dioxide. Right? Again, 3 of these will be fixed, or you'll have 3 cycles to form your G3P. So for every 3 molecules of carbon dioxide, you will have 6 molecules of glyceraldehyde 3-phosphate formed. Right? This guy is going to act as a higher energy molecule used to build sugar. But not all 6 will be used to build sugar. Only 1 is. So only 1 molecule of the 6 total G3P will leave the Calvin cycle to serve in the net gain of a carbohydrate. Right? That net gain will either be 1 molecule of G3P or its equivalent, which is half a molecule of glucose. Right? Again, this guy will be used to synthesize other carbohydrates. But what about the other 5 molecules? What happens to them? Well, they will be recycled. Right? They're going to be recycled into the third phase of the Calvin cycle, which is your regeneration. The remaining 5 are recycled to regenerate the 3 molecules of RuBP, right, which is the starting material or that starting substrate that's used in carbon fixation, upon which carbon dioxide is added to. So these guys, again, are used to recreate or regenerate the starting material for the Calvin cycle. Basically, in order to get this regeneration, what happens is the carbon skeletons of these G3P molecules are rearranged, and then 3 more molecules of ATP are used to allow for this regeneration or rearrangement and addition of components to create 3 molecules of RUBP. So this enables the system kind of to go back to its factory settings, where it's able to accept or receive more carbon dioxide because you have the regeneration or production of RUBP, which can receive and fix the carbon dioxide that enters the system. Now, the ATP and NADPH that are used in the Calvin cycle, they're sustained by light reactions. So light reactions sustain the Calvin cycle by regenerating ATP and NADPH. All right, so what are the nets of the Calvin cycle? Well, for the synthesis of one G3P molecule, right? When I say that, I'm referring to the output of your 1G3P molecule that will leave the Calvin cycle. In order to create this guy, the Calvin cycle will consume 9 ATP, right? Consumes ATP, 3 ATP during the regeneration, and 6 ATP during carbon fixation. And it will also consume 6 NADPH during the reduction phase. And again, that G3P that's leaving the Calvin cycle becomes the starting material for metabolic pathways that synthesize other organic compounds, including glucose, sucrose, and other carbohydrates. So those are the 2 major processes of photosynthesis. Again, starting with light reactions, right, which are going to occur in your thylakoids, and then followed by your second process, which is the Calvin cycle, your dark reactions that occur in the stroma. I hope this lecture has helped you better understand photosynthesis and what's occurring. You can, again, think of your light reactions in the Calvin cycle. They themselves are cyclic, right? So the light reactions are producing ATP and NADPH that will be used or consumed in the Calvin cycle. And in that process, they're broken or converted back into these forms of NADP plus and ADP. And then these oxidized forms will be regenerated into ATP and NADPH in the light reactions, right? So the light reactions are allowing for the supply of intermediates and energetic components that help fuel or drive the Calvin cycle. Again, I hope this explanation has allowed you to understand the concepts of photosynthesis in a better and more meaningful way. And as always, I hope that you are able to walk away with not only a better understanding, but having learned some new information that you didn't know before. This is just part 1 of the photosynthesis lecture component. The second video will enlighten you about the differences between different types of photosynthesis and different types of photosynthetic plants. So differences between C3, C4, and CAM. So you can stick around to watch that. That video will follow immediately after this one. Or you can enjoy whatever you're doing, or if you fell asleep, I hope you have a good rest of your sleep.